I just want to say thank you so much for making the new time work. I will share a few links that I have on my Google Doc with all of you to help you a little bit in terms of figuring out where we could head this conversation. I have to thank Victor Bustos, whether he's here or not, because he gave a previous meeting, which was recorded, on genetics, aging, and COVID-19. And then he warmly recommended to contact Joris, who is here today, to really do a deep dive on this topic with us. And then Joris recommended contacting Albert, who's also here today, to do a deep dive into the relationship between genetics, COVID, and aging. So I'm really, really happy. We have two really strong presentations, and it's super new research, like really off the cuff. I will share all the relevant links with you in the chat. But for now, I just want to give the stage to the speakers. And if the speakers want to kind of share whether they're comfortable or not with being interrupted, then at least we can make it more or less likely. And to everyone else, please feel free to collect all questions, mark with a Q in the chat, and then we'll get to it shortly afterwards. So I just want to say thank you so much for joining. I'll share all of the relevant info, like your bio and your talk description here in the chat. And I'm really happy to have you all here and to see you all again. It's always, it's making my week. So thank you all for being here. And Joris, if you want to take it away, then please feel free to do so now, and I'll share everything in the chat. Joris, welcome.
Good. Thanks a lot. Yes, I'm happy with being interrupted, so don't worry about this, especially when something is unclear. So I have made a presentation about the subject. So you can all see my screen, I hope, now?
Perfect. Yes.
Okay, good. So I want to tell you a bit more about the work I've been doing over my whole career, kind of, which is deciphering the genetics of human longevity. And of course, the main part will be about our latest work. So first of all, as you all know, lifespan has been increasing over the last 200 years. So both in men and women. Here are some survival curves for a Dutch population in the 1850s, 1900s, and 1950s. And as you can see, there's a clear increase. And of course, this increase is mostly caused by non-genetic environmental factors because they were improved over these 100 years. But we also know that part of it is explained by genetics. So before I go into more detail about this, I want you to know what I mean when I talk about certain things in this presentation. So when I talk about lifespan, I really talk about the number of years lived. So we follow the whole curve when we talk about lifespan. However, I always also talk a lot about longevity, and I make a clear distinction between the two because longevity, we consider survival to an exceptional old age. So if you look here, that is the people that are actually surviving to over 90 years of age. And as you can see, this also increased with increasing time. So in the 1950s, there were much more people that were, for example, above 90 years than in the 1850s. So what we know about the genetics of longevity? Well, we know that it's heritable. However, there's a lot of discussion how much of longevity and lifespan is actually heritable. So earlier studies, which were based on twins, show that around 25% is likely due to genetics. However, several recently large pedigree studies now show that it's probably much lower, around 12%, and this may be inflated due to assorted mating. So there are several nice papers on this that you can find here. However, there's no clear heritability of longevity established yet. So what we know is that there is a strong genetic component because we know that people that survive to belong to the 10% longest lived of their birth cohort, we know that it's a shared trait within family. So we know that there is a genetic component for longevity, mainly living to the 10% longest of the population, but we do not know yet how large it is. So there are several ways of studying this genetic component. And today I will talk about what we do when we look at common genetic variants. So these are variants that are easily picked up because they're common in the population. However, you have to be aware that the effect of these alleles, so the allele frequency is quite large, but the effect size is most of the time quite small. So we're looking for small effects of common variants here. So the way that you can study this is using different ways of doing that. So one is using binary phenotype, which we do again when we talk about longevity. So we actually look if the frequency of a certain variant, here indicated with a G, is higher in cases in comparison to controls. You can also do it as a continuous phenotype that you can, for example, do when you study lifespan, and then you see the older people get, the more frequency they have of the G. So it's another way of analyzing the data. So when we study longevity, which we did already for quite some years, these are the latest two big studies, we actually do not find a lot of genetic variants associated with longevity. So how you should interpret this graph is here are all the different chromosomes and all the genetic variants on those chromosomes. And here's the -10 log p-value. And what we consider genome-wide significant, so adjusted for multiple testing in GWAS, is a p-value lower than 5 × 10 to the -8. So once a variant crosses this line, this red line, or here on the top line, we consider genome-wide significant. So although there are several hits here, around 5, only ApoE is really consistently found, also already in earlier studies, and you can see here the axis is actually truncated. It's really significantly associated with longevity. But that was already known from candidate studies that this was an interesting gene to study. So if we zoom into this region, we actually see that there are two independent SNPs determining this effect. The first one is this one, which is also considered the ApoE4 defining SNP, and the other one is located here, which is independent, but also has a larger or smaller p-value, which is the ApoE2 allele. So if we then look at the ApoE4 and the ApoE2 allele in our largest longevity study, we consistently see that people carrying this E4 allele show a decreased increased mortality risk, so actually in a hazard ratio below 1. On the other hand, the ApoE2 carriers consistently show an increased risk of becoming long-lived, so an increased probability of becoming long-lived. So these are opposite effects, which is also known, but it's nice that we could, of course, confirm it with GWAS studies. However, since we felt that we were not so successful studying longevity as a phenotype because we could only get a limited sample size of long-lived people, named the last GWAS was around 12,000. We also looked into other kind of phenotypes that were out there. So there were several studies that were already done, one on parental lifespan with over 1 million individuals and one on health span with 300,000 individuals. And both of these identify way more loci, probably partly because of the sample size, but also maybe because of the phenotype they used. However, when we looked up these variants that they identified in longevity study, we didn't see so much. So only one of this loci, which is this CDKA2A2B locus, was also associated at a reasonable level with longevity. So then I actually contacted the authors of the parental lifespan study, which the first author was Paul Timmers and last was Peter Yoshi. And we came up with a project where we thought, okay, let's combine these different phenotypes. So let's combine the longevity, the parental lifespan, and the health span genotype to see if in that way we can increase power and detect novel loci for longevity. So the first thing that we did is, of course, determine if these three phenotypes, so health span, parental lifespan, and longevity, were genetically correlated with each other, because that would show that we could actually merge them. And when we do that, we see that the largest correlation is there between lifespan and longevity. It's about 0.8, but also the other two are reasonably well genetically correlated. The smallest correlation is there between health span and longevity, but it's still above 0.5. We then decided to look if we split now lifespan into people living to an age between 40, 60, 60, 80, or 80 plus, do we then see a stronger correlation with the older lifespan? But that's not what we saw actually when we looked at longevity. So it seems that the high correlation is not caused by the fact that it's mainly driven by the elderly individuals when you look at parental lifespan. What is more likely the case, the genetic correlation is driven by the shared genetic architecture of these three traits. Because if we compare the genetic correlation of all these three traits with disease-related traits, we actually see that there are a lot of traits that are shared. The show is shared genetic correlation with all traits, namely, for example, coronary artery disease, which is quite strong. And stroke is also, of course, related to that, and type 2 diabetes related traits. However, we can also see that there are some specific traits that are specifically associated with only one of the traits we studied. Take, for example, cancers, which are really specific for health span. And that's also because health span when we looked at health span, or the people looked at health span, they took into account cancer as well. And since cancer is one of the first occurring diseases, you see an effect in health span, but not so much in lifespan and longevity. However, for longevity, for example, we could see a genetic correlation with Alzheimer's disease, which we could not see for the other two. But the reason that they are probably sharing here a high genetic correlation is because they are all related to coronary artery disease and type 2 diabetes. So then we combined the three phenotypes, and then we did the GWAS. And when we did this, these are all the loci that we identified, in total 24. So the majority of these had already been found to be associated with one of the traits separately. And there were 10 loci, so which I highlighted here, which were associated with all three traits at a nominal significant level. So by combining them, they became genome-wide significant, but also at a nominal significant level, they were associated with all three traits. So we found those the most important. There are several known ones, again, for example, ApoE, the CDKN2 locus, but also several new ones. Like now, for the first time, we could finally identify FOXO3, but there were also 4 other ones that we had not seen before. So when we zoom in into these regions and look to the variants that are within these regions, we wanted to determine, okay, with what kind of phenotypes are they associated? And of course, not unsurprisingly, we again find an association of most of them with cardiovascular related disease, but also with several other things like metabolic phenotypes and immune related phenotypes, and some even also with cancer. There's one locus, the FOXO3, which is also related to smoking. So this was something that we could expect based on what we already saw earlier. But then the next step, we were thinking, okay, so what kind of genes are actually influenced by all these loci? So what we did is a so-called EQTL lookup, mostly in blood. We also used other tissues, but most of the results are in blood. And then we looked, okay, if we have the genetic variants, with which genes are they associated? So what you would expect is that most of the genes most of the variants show an association with one of the genes that is close to them, which is called a cis gene. For example, you nicely see that with FOXO. You see this with this locus, the CDKN2, and also, for example, with, there are several that, FGD6. So it's clear that probably the genetic variant influences really this gene within the locus. However, for some genes, it's less clear. Some of them do not have cis genes, or some of them have cis genes that are actually a little bit further away. So the genetic variant itself is located within this gene, actually within the intron, but it is the question if it really influences this gene or if it actually influences another gene within the locus. We can also do that in trans. So in that way we can see, are there other regions in the genome that are influenced by this genetic variant? And that showed actually quite some interesting findings. First of all, when we look at the genes that we know show an increase or decreased expression with age, so there was a large genetic study on this, which looked at the age-related expression of genes in blood. We show that the genes that are highlighted here all show an age-related increase or decreased expression. And interestingly, especially this locus, this link RNA locus, was associated with several of the top genes within their list. So even in their top 10 of age-related genes in blood, some of them are here. I think three out of the top 5 are in here, which means that this really seems to be a gene that is a locus that's influencing gene expression in blood, age-related gene expression in blood. And when we zoom in a bit on what kind of genes are influenced, it mostly seems to be immune-related genes. On the other hand, what we also identified is that some of the genes are associated already with lifespan extension in model organisms, namely FOXO3, for example, which has been shown to associate in different organisms, IGF-1 receptor, and this SNCA locus, which is coming from the worm. So this also confirms that by doing these genetic studies in humans, we are able to detect variants influencing genes that are also doing something in model organisms. And then we decided to see, okay, are there specific pathways that are enriched in our gene set? So we took all these genes that I showed before and then did a kind of enrichment analysis. And when we do that, one really striking result was that we found heme metabolism. And as you can see here, the genes that are driving this are mostly within the LDL receptor locus, although there's also one in the FOXO3 locus. So we thought, okay, this is an interesting finding, but can we actually believe it? So we decided to do also use a different technique, so Mendelian randomization, to actually see if heme metabolism was indeed involved in our new healthy aging phenotype. And the results you can see here. So how you should interpret this. So this is the effect of the variant on, for example, serum iron, and this is the effect of the variant on lifespan. So as you can see, there is a trend, and it's also significant that when you have SNPs that increase serum iron, they're actually associated with a decreased lifespan. And you also see the same when you look at other iron-related traits like ferritin or transferrin saturation. So this gives us some evidence, more evidence that this actually seems to be a real relation, so that the serum iron-related SNPs seems to have an effect also on lifespan. So the conclusions of this study are then that even that by combining the three phenotypes, we actually overcome the problem of GWASs of longevity, namely that we have an insufficient sample size to detect new loci, and we were able to detect several novel loci. And then we identified novel genes and pathways that we now want to further test in model organisms and cell models. So with this, I would like to end, but not, of course, before thanking certain people. So maybe Paul and Peter, for which were my co-authors on the study, and and Paul mostly did a lot of the work and created all these nice figures. And of course, all the other people here that have been involved in all the longevity studies that we had done before. So, do you have any questions?
That was a super fast presentation. I think we have an immediate question now. Kejun, you want to go for it?
Yeah, thanks. Really interesting stuff. First of all, I want to make sure I understand what's going on here because I'm not a genetics person. When you're looking at these SNPs, is that basically telling you the rough magnitude of how much a certain gene is being expressed in an individual at that time, or is it just telling you the presence of a particular allele? It
only tells you the presence of a particular allele, but with the EQTL studies, we can actually say if the SNP influences the expression of a gene, but we do not know the expression over time. So it's just if the variant is present in your genome or not.
I see, because wouldn't it be, presumably if something's presence matters, then its level of expression would also matter, I would think. Like if it's present but not expressed, who cares? No?
Yeah. So I mean, of course, the variants are not expressed, but we think that they influence indeed expression of genes. It could also go a layer in another layer. It could also express that it directly influence proteins without influencing expression of genes, or it could influence epigenetic the state. Right. So these variants can do different things.
Got it. Complicated as usual. So that brings me then into my real question, which is kind of two or three things bundled into one. One is, have you been following some of this new glycomics stuff? And do you think that either these genes are affecting glycosylation or some other epigenetic thing, or they get themselves, if they code for proteins, that there's some downstream pathways that are still important? And then regarding especially the ferritin and the iron loading, are you aware of some of these evidences that actually decreasing your iron by, say, donating whole whole blood and things like that can have a potentially positive effect on certain health and lifespan issues? You're aware of that. I see you nodding.
Yeah. Yeah. So regarding your first question, we haven't. Well, what we have done, of course, is looked if these variants have been associated with glycan-related traits that have been published. We couldn't find that. So we don't think. Well, at the moment, that's not yet happening. We do not know if the genes that are influenced by glycans. I guess that's probably the case because a lot of genes are influenced by glycans, but we do not have any evidence yet that the specific variants influence the glycan status of these genes, for example. And regarding your second question, yes, I'm aware of that this kind of studies. And I also want to highlight that what we found actually, if you, I maybe go back to my screen, maybe that's easiest. Then I can explain it. So if you look into the data here, actually the variants that we identified are really strongly influencing iron levels. So people that are carrying these genes have extremely high levels normally, especially for example, this HFG gene.
And what's
the
beta?
HFG variant? Sorry?
What is beta, or what is the y-axis?
It's the effect on lifespan. So what we see is the higher. So this is actually, so if you have a minus here, it means you have a decreased lifespan. If you have a plus here, it means an increased lifespan or healthy aging. But what you should realize is that, of course, this data is created based on people which have variants that clearly influence iron levels. And these variants are really strongly influencing iron levels. So likely when you do not carry them, you're protected. But it's not said that if you have a normal iron level, which is not influenced so much by these genes, it would also be good to decrease your iron. It's just saying that if you have really high iron, like these people, that leads to a decreased lifespan. And those people might benefit from lowering iron, which would also make sense. But it doesn't say that people that have a normal iron level should also always decrease it, because that we cannot tell from this kind of studies.
Yeah, so regarding that, is Nicolina on the line here? Oh, fantastic. Nicolina, do you mind if I mention that potential informal investigation we were discussing, or should I not?
Yeah, absolutely. So to answer your question, well, so far Gordon would say that it's not directly related, but I'm interested in the therapy to see if that has any effect, because there is other side. So if you're a senior and you're donating blood, you could be doing damage to the immune system. But if it's somebody who really has an overload of iron, or it's affecting him negatively, and that helps with other things, that could improve glycan age. So absolutely put it forward. If we have 10 people, it's an interesting case study. And yeah, we will be super keen to do it. So,
so let me mention what we were talking about here. Nicolina and I sort of had this idea. I had gotten a little bit seduced by this dumping iron kind of hack, and I wanted to see what it did to me. And so pre-COVID, I gave blood a few times, and I was just tracking it and seeing how I felt. There was nothing formal. And then I stopped doing it. And then because of the lockdown that we're still under here. And then when Gordon and Nicolina were presenting their stuff, and they mentioned iron, I believe, we came up with this idea of maybe recruiting a small group of 10 people from this cohort here, or maybe an extended Foresight group. And if we could find 10 people who'd be willing to commit, we could do glycan age before, and then we could donate blood at a certain frequency for a while, and then we could do glycan age afterwards. And maybe we could also pile on some other tests or something like that. And that would be really fun to see if it's causative, right? Because we don't really know if the iron here in your graphs is an indicator of something else, or if it's causative in reducing lifespan, correct?
Well, the idea is that Mendelian randomization, what we did here, is proving kind of causality, in the sense that we know that the variants are influencing both traits, so it's a direct effect. But like I said, I mean, the variants that we are using for this are the ones that are really influencing iron at a strong level, so that in normal people, most of them will probably not carry them, and if and they will not have this massively high iron levels. So you should be aware of this, that we cannot say what it does.
Is this an example of? Is this an example of Mendelian randomization? This study right here.
This is Mendelian randomization, yes. So my co-author did this. I don't normally not running this, but he did it, and this is really how the Mendelian randomization example. Yeah.
Okay, I've overstayed my welcome. I'm shutting down.
Well, I'm hoping I didn't lean too far out of the window in saying that if anyone's interested in this, then they should contact Kejun or Nicolina, and then maybe we can add you to that group of the 10 test cases. Is there anyone else who has a question that's directly relevant to Joris' presentation? Otherwise, we'll move on to Albert's presentation, and then we'll do a discussion later. No one? Okay, then great. Joris, it would be amazing if you could stop sharing the screen. Otherwise, I'll do it. Okay, good. Thank you so much. Okay, well, thank you so much, Joris. That was fantastic. You can tell me afterwards if any of this is too pre-pub to publish anywhere, but I think you base most of it on the research that's already out there. Albert, thank you so much for joining. Thank you so much on kind of like giving us the COVID-19 relevance, which is definitely, I guess, the impetus for how this group was started. It's definitely not the only reason why we talk about this, but I'm super curious to see kind of like what findings you've done. And I'm just seeing that Vadim is connecting his audio, who was super excited about having you. So I'm super glad. And maybe even Vadim has a few questions that could point the group in the right direction. For now, I'm super happy to have you here. Please launch your presentation. I'll share all the relevant information on you in the group chat. Welcome, Albert.
Okay, thank you, Alison and Joris, for having me here. I'm going to share my screen. So just to briefly introduce myself, I'm Albert, and I'm a PhD student in Harvard School of Public Health. And I joined Vadim Gladishev's lab in earlier this year, and where we study the mechanism of aging and longevity. And today I'm going to introduce our recent work on aging and COVID-19, which is actually a build-up on Joris' work. And I'm going to talk about the evidence we found at both genetic and phenotypic level that point to the causal relationship between aging and the risk of COVID-19. And here I'm showing you a piece of data from a recent paper from our lab. It's representing the relationship between the COVID and aging. And you can see it's obvious that both the case number and the case fatality rate of COVID are increased with age. And although the case number the trend is not consistent here, but it still shows a strong link between aging and COVID-19. And based on this, there is a tempting idea of using various anti-aging drugs to prevent or even treat COVID-19. And in fact, there are multiple clinical trials has been proposed. However, it could be dangerous to just rely on epidemiological evidence because the observational data are prone to confoundings and various bias, and therefore it's an unreliable indicator in predicting novel intervention. So to actually support the idea that repurpose the anti-aging drugs for COVID-19 prevention, a causal relationship between aging and COVID-19 need to be established. So when we're talking about causality, the randomized clinical trial is the gold standard on establishing causal relationship. So in randomized clinical trial, we first sample a group of people and we randomize them into intervention and control arm. And this is actually the key part because as long as the sample is properly randomized, the confounding variable here will be equally distributed in both arms. And therefore, the result here will be free of confounders. And then to test causality, we can just modify the exposure we want to test and check the outcome. And the problem of the clinical trial is very obvious here. We're testing on actual people, so there might be ethical problem, and it's very expensive, it's time consuming. And there are numerous drugs that at this time are waiting for the trial for COVID-19. So to prioritize the anti-aging drug here in clinical trial, we will need additional evidence. And here comes the Mendelian randomization. As also introduced by Joris, Mendelian randomization is a genetic approach that uses genetic variant as instrument to test the causal relationship between the exposure and outcome. The idea is very similar to the randomized clinical trial. So first we genotype a population, and because the genotype is assigned randomly when passed from parent to offspring, so we can consider genotype as a naturally randomized, just as in randomized clinical trial. And therefore, the population genotype distribution should be unrelated to any environmental confounder. So we can use the genetic variant that lead to, for example, slower aging rate, and to test the causal relationship between aging and COVID-19. And Mendelian randomization can provide valuable insight into causality when it is infeasible to perform randomized clinical trial or before the trial is performed. And this is actually what we are doing here. And here we use a multiple instrument Mendelian randomization approach to investigate the causal effect between the aging and COVID-19. And here I use 11 different COVID-19 related traits that obtained from different resources as outcome. And I use 4 traits that, and the 4 traits here we use as exposure are exactly the 4 phenotypes that Joris just introduced: the health span, lifespan, longevity, and this combined trait. We immediately use this meta-analysis result when we notice Joris published this. And in this figure, the bar here are showing the odds ratio, and the black means the P is more than 0.05, and the orange means it remains significant after corrected for multiple testing. And from here, we can see that the genetics of both longer lifespan and longevity are significantly associated with a lower risk of the COVID-19 infection. And you can see it more clearly from this plot right here. The dots here are the genetic variant that we use as instrument for lifespan. The x-axis is the effect of this instrument on lifespan, and the y-axis is the effect of this instrument on COVID infection. So you can see the genetic variant that lead to a longer lifespan are also lead to a lower risk of a COVID infection. And this effect is very dramatic, actually. For every 10-year longer genetically predicted lifespan or just parental lifespan, the risk of COVID-19 will decrease by 68%. And it is also a little bit surprising that health span doesn't show any protective effect at all. And this result might suggest that the strong protective effect of longevity against COVID-19 should be explained by a slower rate of aging itself, instead of the delay of the appearance of the age-related disease. And to further investigate whether this protective effect is explained by any age-related disease, we look at the additional 4 diseases that explain the most of the lifespan variants in lifespan GWAS, including the late onset Alzheimer disease, cardiac artery disease, smoking, and type 2 diabetes. And here, only the late onset Alzheimer disease show to be a risk factor for COVID infection. And the effect is actually kind of small, and it's less significant compared with the lifespan and longevity. The odds ratio here is 1.13, means that there is about 10% increase of the risk of being infected for people with Alzheimer. So it's hard to believe that the strong protective effect of longevity we observe is mediated by Alzheimer disease. And together with the result of health span, we believe that the protective effect of longevity should actually not be explained by this disease, but by decelerated biological age itself. And to get the full picture of the relationship between the disease and COVID-19, we did another Mendelian randomization using 22 common diseases as exposure from JERA cohort. And none of them shown to be significant after corrected by multiple testing, but they still giving us some information. So among the 11 COVID traits we use as the outcome, we can classify them into two categories: the trait that representing the COVID incident and the traits that representing the severity of the COVID case. So you can see here the disease like dyslipidemia here are risk factor for the COVID infection, and the disease like hypertension are the risk factor for the severity of COVID. And there is one additional very interesting trait here called disease count. It represents the number of the comorbidity that is affecting the individual. And this result here is actually feed the observational data that indicating people with more comorbidity are more likely to develop severe case and eventually die from COVID-19. And then we move to the evidence at phenotypic level. We use the phenotypic measurement in the UK Biobank cohort that collected prior to the pandemics. And we use this phenotypic measurement. We applied multiple biological age model and calculate the biological age acceleration. Then we ask the question, does the people who with higher biological age acceleration are more likely to get COVID and die from it? And the result is exactly what we expected. For phenotypic age, which is the measure based on blood chemistry, we see people with increased phenotypic age acceleration are more likely to be tested with COVID and die from it. And also DOSI is another biological dynamic organism state indicator, which is computed from complete blood count. And you can see same pattern here. And as well as physical activity, which is defined as the number of steps per day and at a negative log scale. And we also see that less physical activity will lead to increased risks of COVID in the future. And this pattern is actually shows no dependence on the appearance of comorbidity. For example, for phenotypic age, we see a similar pattern in people without comorbidity and people with comorbidities, and same for DOSI. So this result again suggests that the effect is not fully mediated by disease, but instead by aging itself is the main driver here. And lastly, we calculated the predicted risk of COVID with accelerated biological age. Again, the bar here shows the odds ratio, and the orange means a significant after bone protein correction. And you can see as consistent with our genetic evidence, the acceleration of biological age increased the risk of being infected. And the risk is increased by around 30% with every 10-year acceleration of biological age measured by phenotypic age and DOSI. And the association between case fatality rate and biological age acceleration is actually less significant. You can see it's just reached the nominal significance and non-significant for DOSI. And actually, the case fatality rate is only predicted by accelerated biological age in healthy people, but not the people with comorbidities. And we also found that people who walk every 1,000 more steps per day will have 5% less risk of being infected by COVID. And again, note that this is measured before pandemic, and it doesn't sound like a good idea to just randomly working outside at this time. So I think it's better if you want to do it, better do it at home, like walking around in the living room. I personally do it a lot. And to summarize, at genetic level, we found that the genetic variation that lead to longer lifespan is causally associated with lower risk of COVID-19 infection. And the protective effect is likely to be mediated by the slower rate of aging. And at phenotypic level, we found that the biological age acceleration measured by phenotypic age and dosage and physical activity are associated with future incidence and severity of COVID-19 infection. And again, this association is not dependent on comorbidities. And together, our findings suggest a causal relationship between aging and COVID-19, which directly point to the possibility of using anti-aging drugs to prevent COVID-19. And yeah, and that's for acknowledgement. This work is co-led by Vadim Gladyshev and Shashan from University of Edinburgh. And Ranjai from his set is my co-first here. And this work is also done by collaboration with Peter Fedoshev and Timothy Pyrakov. And we also need to thank Deelen Lab and Joris Lab and many other labs for making the GWAS summary statistics available, and also the NIH GRAS and HGI for timely release of the COVID GWAS. And thank you. And I'm going to take questions.
Wow, fantastic. Thank you so much for sharing. I think there's a few questions that are lined up. I will take Carl first and then yours second. So Carl, go for it.
So the way you presented the phenotypic age as a risk factor was fine. I'd like to see if it's easily manipulable in something that's more easily understood by lay people. So you looked at the phenotypic age acceleration, meaning, you know, to what extent was your phenotypic age higher or lower than the chronological age? So showing that difference is a risk factor. Essentially, correct me if I'm wrong, essentially shows that phenotypic age is a better predictor of COVID-19 risk than chronological age.
No, we actually cannot say that because we only look at the phenotypic age acceleration. So this means that if you have accelerated phenotypic age, which is your phenotypic age minus your chronological age, and we only look at the acceleration here. And our result here are.
So if chronological age, if risk goes up with chronological age, and acceleration also shows risk, then it seems like phenotypic age must therefore be a better predictor than chronological age.
Yeah, you're right. Yeah, actually.
So can you directly look at that as a comparison in terms of which one is a better predictor, and then quantifying the extent to which the phenotypic age or any other measure of biological age is a better predictor than chronological age? Perhaps one way to simply visualize that that might be understandable by lay people is to take the slides at the beginning where you showed the incidence risk and the severity risk by age group, and instead plot those same slides by phenotypic age instead of chronological age. You know, it would be not, I think, what I think would be useful here, I understand one of the motivations for this work is that this justifies the idea of looking at anti-aging treatments as potential prophylactics for COVID-19. But quite frankly, I think that that's actually, you know, we just don't have enough good anti-aging treatments. I think that's actually not that great a line of work. But from a public relations point of view, actually showing people that this easily available measure of biological age is a better predictor of risk factor, you know, especially phenotypic age, which you can actually compute from simple blood tests, right? That is a real, like, if that could get out as a message, you could have a little website, type in your 5 values from your blood tests, and it'll tell you your phenotypic biological age, and that actually, and here's the risk factor bar chart versus age. Like that would be a really interesting, you know, maybe Keith can comment here, a really interesting public service website, I think. And here's the research that shows these are the risk factors, blah, blah, blah. So, I don't know, comments on that.
Yeah, I agree. It's a good idea, actually.
Yeah. Oh, Keith or Joris, do you want to chime in?
Yeah, I was basically just going to say, I agree with what Carl was saying. Anything that can significantly message to the public in a very easily understandable way the value of understanding your physiological age in relation to COVID would be a public relations coup for our field. So I agree with that, Carl.
Yeah, I also agree. And I think it would be good indeed if you show in all your graphs also the effect of age itself for the comparison. I think that would be a really good idea. So I actually wondered, I had a question about your differences between lifespan, longevity, and health span, because we know that health span has some limitations the way it is defined. And it seems that your phenotype is strongly associated, or the COVID phenotype is strongly associated with Alzheimer's disease, right?
Yeah.
While we know that in the health span GWAS, it's based on the occurrence of the first disease. So there is not so many people which have as a first disease Alzheimer's disease. So that's really underrepresented. And actually the way we know that this is happening is because when we look at the ApoE locus, it's not so strongly associated with health span. It's very strongly associated with the other phenotypes, but not with health span. So this might explain why you do not see your effect so much on health span, but it's mostly on lifespan and longevity, because Alzheimer's disease plays such an important role there. So it's something that you can take into account and maybe look into it a bit deeper to see if that's indeed the case.
Thank you.
All right, that's great. Well, does anyone else have any questions, comments?
Yeah, I have a question.
Yes, go for it.
I had a question in terms of, there's some underlying mechanism of aging that's still sort of mediating this effect. And do you have any theory about what that is? Is it something related to Alzheimer's disease, which could be either neurological or having to do with cholesterol and inflammatory processes? Or is it immune aging? Do you have any data or theories about what that might be?
Yeah, we actually think it's because a better immunological profile in people in like centenarian or people who live long. And there are actual studies that find that the centenarian have some special kind of immune cell, and they also have less inflammation. And actually, it's actually heritable. Like the offspring of the centenarian also have a lower level of inflammation. So I think it's might mediated by the immunology.
Yes, I see a question.
Okay, so we've been talking now here and elsewhere around this issue of the like the genome age predictors and the phenotypic age predictors and the epigenetic age predictors and then these other things like frailty. And then it raises the question for COVID and just about anything else. Like if you could factor all this stuff out, does biological age is it even causal, or is it just a proxy, rough proxy for these other molecular and informational situations that are correlated with biological age? And so I guess what I'm trying to say is at some point it's going to be very interesting to kind of map out the two-dimensional probability distributions once we have enough data, like chronological age versus biological age, and see how things vary in that space, because it may turn out once we get our act together that in a sense biological age is it's irrelevant. Like lifespan, ideally, if you think about it, maybe for the longevity movement, we want lifespan to be independent of chronological age. We want it just to be affected by various causal, molecular, and lifestyle mechanisms. And so I think this will be very interesting. It's sort of almost like a goal of all of this is to get so much data from epigenetics and biological insight that actually the chronologic age is no longer a predictor. Like if you took out all the frail people and all the people with morbidities and bad genes, there'd be no age dependence on COVID death. Anyway, just a rant.
Yeah, makes sense. Thank you.